Recording in progress.
Good evening. Welcome to SVN3E Environmental Science. My name is Wendy Espana, and this is our last night on Lesson 19. We're going to finish off Lesson 19 tonight, and then we have one more lesson left, and that's Lesson 20 in the course. Lesson 20 is a bigger lesson. It's going to take us a few nights, and then hopefully we'll have a little bit time at the end that we can start doing some review before we get prepared for the final exam for the course. Okay, we are going to start down on page 9 of 15. And let's just remember our lesson 19 learning goals. Students will be able to describe the 3 types of biodiversity, describe the value of biodiversity to ecosystem strength, and describe common methods of measuring and monitoring biodiversity in various habitats. And tonight, the common methods of measuring and monitoring biodiversity is what we're going to concentrate on. Okay, so if you just give me a minute here, I'm going to quickly go over to our textbook questions, and we will just get that ready on the screen here. And there we go. Hopefully you can see that. And we are on 6.2. And 6.2 in the textbook is measuring and monitoring species biodiversity. And you can see there that we have a chart there right at the top of the page. Sorry, we're on page 162. Page 162. And you'll see that there's a table, and it's got examples of different types of animal species identified. And you'll see there's a pie chart, and the big portion of that pie chart there we can see on page 162 is invertebrates. That means they don't have spines, right? So we've got insects, spiders, worms, and you'll see in there is 1,359,367. That's how many species they found of insects. You can see that insects make up the majority of life on our planet. Something that's not on there are viruses and bacteria, and they are huge too. I do know that they take up a huge amount of species on our planet as well. There's also mammals, 5,487. Amphibians like frogs, 6,515. Reptiles, those would be like lizards, crocodiles, those types of things, 8,734. Birds, lots of birds, 9,990, and fish, 31,153. And that's where we get the 1.9 million different species that have been identified. So the organisms without backbones, without spines, the invertebrates make up the majority of those species. So what we need to do is we need to be able to come up with ways to sample biodiversity in an ecosystem. And in particular, we want to look at how we sample biodiversity over a period of time, right? So we check in, we see what the biodiversity is at a certain time, and then we keep track of that, and we just keep checking back in. And every time we check back in, it gives us an idea of how that ecosystem is doing. Okay, we're really interested in if something is changing over a period of time. Okay, and especially if it's changing quickly, that's very alarming. But even over a long period of time, a longer period of time, we really are interested in how the ecosystem is being affected. Remember last night, we talked about some of the factors that can affect an ecosystem: climate change, pollution, global warming. All of those have an impact. And we looked in particular at polar bears and We've been looking at them because they are of particular interest to Canadians, and especially Canadians in the north. And looking at what's happening as the temperatures get warmer and the sea ice melts, and what happens when they can't hunt and get the food, the proper food, not just eating berries and things on the land they can find, but actually being able to go out onto the sea ice and hunt with seals, which is their what they should be eating. It gives them the nutrients, the fat that they need to survive. All right, so we want to keep an eye on organisms and the biodiversity of those organisms in an ecosystem. Okay, now it is difficult, if not impossible, to go into any ecosystem and start counting things. Uh, this is particularly true of these invertebrates listed here. We know that invertebrates, smaller animals, amphibians, fish, those are ones that are very affected by smaller amounts of pollution, toxins, right, in an environment, in an ecosystem. And so these would be the ones you would want to be counting. So you have to come up with ways that we can see how many of a species are in an ecosystem within a particular habitat, maybe, and sampling. It's the way that we can take the number of species that we can count in a very small area and then estimate how many there must be in the whole habitat, in the whole ecosystem. So we're going to go through some of the sampling methods. I think it's kind of interesting because this is really science, right? This going out into the field and collecting samples and being able to make estimates based on that is really fascinating scientific work, if anybody's interested in that. Biologist is a career here in this area that you would work for different, probably for the government, the Ministry of Natural Resources, and you would go out and do this sampling and present your results to the government to let them know if they should be, concerned about, we were just talking about the number of polar bears, maybe a quality of those polar bears. Okay, quadrant sampling. It's a common method for measuring biodiversity. What you do is you take a gridded plot. A quadrat is the gridded plot. And there's a picture on the next page we'll show you in just a minute of what I'm talking about. And they place that in the habitat or the ecosystem that you're studying. And then you can identify, measure, count, record your data about the species that you find in the quadrant. Okay, they can place randomly. Different ones place them different ways. There's lots of different ways to place the quadrants. And then you go, you take that wherever it's lying in your habitat and you count within that quadrant. They can be square, rectangular, circular. There's different types of quadrants. Okay, we're on page 163. And we can see at the top right-hand side that there is a quadrant. There it is. It's that rectangular shape with the grid. And so what you would do in each one of those squares is you'd have a look how much grass, plants, if there's plants in there, animals, what you've got in the soil. Perhaps you would dig down into the soil. Okay, so they usually try to keep the quadrants the same size and the same shape. So if you're going to use that rectangular one like that one in figure 6.6, that's the same one you would use throughout the habitat that you're studying or the ecosystem you're studying. Then what you can do is compare how many species are in the area by comparing the different quadrants. And also you can count how many. Abundance means how many. So researchers can calculate the abundance of organisms in the area. You can use the number you find in each quadrant and then the size of the study area, and then you can measure out how many organisms are in the species from those samples. Okay, there's two types of quadrant sampling. We have passive or active. Okay, basically it means in passive sampling that you don't remove any of the organisms, plants, any animals. They are removed from the quadrant. So you go down, just like that gentleman's shown there. You go down to the ground, maybe that's where you're counting. And they right there at that site, they do the counting and the studying. Maybe they take pictures. Okay, you could take pictures, videos, whatever you need, but you don't disturb the plant life or the animal life. With active sampling, you take the organisms and plants that you find in the quadrant and you collect them. You bring them back with you, put them in, you package them up, you label them, you know where they came from, and then they're taken back to a lab. And then they do all the research that needs to be done. And then they can take and estimate the population afterwards. Usually, if it's something that they want to measure the size of the species, maybe how big the population is in the area, they usually use active sampling. Actually go in and collect an amount. You'd want to be careful, of course, very careful that you're not disturbing the ecosystem, you're not causing any harm to the ecosystem, whether you're doing passive or active sampling. Okay, there's also something called line transect sampling, and that's shown at the bottom of page 163 in figure 6.7. It's another method that can be used to study biodiversity. And you can see in the picture, it explains it right there. You use a long string, a rope that goes across the study area. And then you can collect data along the line. Maybe there's certain every, so many meters, you're going to stop and collect data. And you can count the species that's over, under, touching the line. And then you record and count them. And then you could also record species you don't see but expect to see. There's all sorts of different things you can do with line transect sampling. So if there's a lot of different geographical differences in the area, this might be a good method. And the line could be over a river, through a forest, in a field. There's lots of different ways line sampling can be used. And you could also have a quadrant alongside the line transect. So what could happen is you could lay your line down of where you want to have, it could be a good sites that fall in the line to be able to measure and collect your samples. And then you would lay that quadrant down and you would use the quadrant to measure out exactly where. So you'd have a bigger area that you could actually collect your data from. And it would also be another way for you to collect data. Okay, so another one is on the next page, page 164, and it's aerial photography. If you have a bigger area that you want to know about the biodiversity in a bigger area, aerial photography is a great way. Usually they use black and white, infrared, color, film even. So infrared picks up heat, right? It's a way that we can see heat. And so what it can do is it can measure where animal in a photograph sees more than we can see with the human eye. And so it's another way of getting beyond even what we can see. So it's a great way to collect more data. In figure 6.8, we have an aerial photo of Weemup Canyon. And that's Lake Superior way off in the distance there. I don't know if anybody's been to Weemup Canyon, but it's beautiful, beautiful. And it would have, I know that there's special quality of Weemup Canyon. If you look in Weemup Canyon has an ecosystem in the bottom of the canyon. Just see if we can see it here. Right. I'm going to put that on the screen. Actually, we're just going to take a little go over for a minute to just going to go and show you. There's cool really cool area. There we go. And it's got, I'll show you here. Just getting it on my screen. If you give me one more second, I'll find it for us. There we go. Okay. I just gotta find it on my screen. Okay. Weemup Canyon ecosystem. Here we go. Sorry, it's taking me so long. Okay. So we see here the Weemup Canyon ecosystem is unique because its floor has subarctic tundra, supporting plants that are typically found 1,000 kilometers further north, while the top of the canyon is boreal forest. So when you get to the canyon, it's really great. This nice photo there of the canyon right there. You can really see that it's quite high, and there's a lookout if you go there. It's a provincial park, so you can just drive there. It's on the other side of Thunder Bay, and it's got a walkway where you can walk out, you can look down, and you can see in the bottom of the canyon is an subarctic ecosystem. And at the top, it's just the one that we're used to. And the difference is quite drastic. The microclimate, it's called, is the Shan Canyon's depth and sheer cliffs, cooler, sheltered environment on the floor, allow Arctic and subarctic plant species to survive down in the bottom of the canyon. The top of the canyon features typical boreal forest vegetation, and the bottom contains subarctic tundra. So there's 1,000 kilometers of distance, and I'd say that's not 1,000 kilometers of distance at all. That can't be right. It says separated by 1,000 kilometers. Oh, usually it's separated. Sorry, that wasn't making any sense to me. Usually that plant life there in the bottom of the canyon's found 1,000 kilometers north. I get it. Sorry. The area supports a variety of bird life that can be seen when you're walking, and it was formed by glacial activity. And the glacier moved through there and caused that canyon. So yeah, that's a cool one. So you'd be really interested in looking maybe at the bottom of that canyon, but you would really want to be careful not to disturb it, not to go walking down there, because it would be a very special ecosystem. You wouldn't want to disrupt anything. So instead, what you could do is do aerial photography and take a picture of that. Okay. So we're going to go back to our textbook. And just give me 1 second. Get back on the screen for us. So if we look at, now we're back on page 165. All right. The Lincoln Index and Capture Mark Recapture sampling. So another type of sampling scientists use, biologists use. Since they tend to be rooted in place, plants and trees are fairly easy to identify and count. Not so true, right? You can just go in and put your quadrant sample down and you're all set. But what about animals, birds, and fish that roam over long distances? In these cases, researchers often use a method called the Lincoln Index. Okay, so it's an index that's based on capture, mark, recapture sampling. Okay, so there's lots of different capture methods. And table 6.3 and figure 6.9 show all the different capture methods. So the first one in table 6.3 is aquatic dip nets. So you can see down on the bottom, they show the biologist there at the bottom in figure 6.9, and they're using an aquatic dip net. And they're in water that they can walk in, and you walk through and you collect insects, organisms, worms, clams, that type of thing. But it would be very shallow water. Mist nets is the second type of net. You can see in the bottom picture on page 165, there's somebody there with a mist net. It has fine nets that are attached to poles, and they block birds, bat flights. So those are how they catch birds and bats. It's difficult to see, so the birds and bats don't see them. And then they fall into the net, and then they're quickly removed by researchers if they're doing passive sampling, and will allow them to be gone as soon as they identify them. And with active, they'll put them in some sort of a cage or something, and then they take them back to be identified and counted, etc. The third one is the helicopter net gunning and darting. This is one where they're trying to, study bigger animals, wolves, grizzly bears, polar bears, etc. Using helicopters, the nets are dropped over the animals. They're shot with tranquilizer darts, and they temporarily stop them. They get out, they measure them sometimes, they tag them. We're going to talk about that in just a couple of minutes. And that's a way they can do a quick count and check, check the animals, species, count species, etc. Live traps aren't shown here, but there are lots of live traps. The idea is that you're not harming the animals in the trap. You're just trapping them to be able to get the information you need. They're good for studying animals that are very shy, nocturnal animals. Nocturnal animals are animals that only come out at night. And I don't know if anybody's ever watched the show Alone. Alone is a show about people who have to survive in the wilderness. And they have cameras that are have infrared, and they can see at nighttime. And when something they're what's the word I'm looking for? Not action triggered, but if there's movement, it turns on the camera. And so it's really neat. You get to see what's happening at nighttime. You get to see ox, break into a trap and get right back out. You see, and all sorts of animals. And so those are associated with live traps. And these ones are not to kill animals. They are to just study them. And so the idea is not to harm the animal, but be able to at least see what they're doing. Once they have the organisms captured, they are marked, and then they're released to go, back to where their population is. And then they get a second sample from that population. And it can be hours, weeks, or months later. The individuals in the second sample are not marked, but you may have gotten some from the first sample. And so you think, okay, the proportion of marked organisms in that first sample must be the same as the proportion of marked organisms in the second sample. And so they use this little, it's, what's the word I'm looking for? Formula. And it will figure out how much the total population is once you've done your measurements. So it's a way that they can do the estimate of how many organisms are in that population living there. And remember, we said that was what they were trying to do, right? Estimate the population. There's a few things you need for this type of sampling to be effective. You have to have a closed population, so you can't have individuals entering and leaving the area that you're trying to study. And the time span between getting your samples has to be pretty short compared to how long the animals you're living. And the ones that you mark have to go back right with the rest of the population before you take a different sample to make sure you get a good mixture, right? The next one is one that most people probably know about, and that's wildlife tagging and tracking. There's lots and lots of different ways nowadays that they can tag and mark wildlife. Sometimes they just mark them and then they, if they find them again, they've been marked and they know where they were, and they can get all the information about maybe a migration route, something like that. They take and put on their leg a little metal or plastic ring, and it's got numbers on it and letters, and those tags identify the bird or the animal or whatever it is that's been tagged. And then they can share with other scientists that they found it and give the information, or maybe they themselves found the same animal. And then you can compare migration patterns, right? Survival rates, that type of thing. How long has that particular animal been, you've been studying it? And you can watch where it goes. There's another variation on this. This is a very simple one. Well, I don't know how simple it is to collect the animal and put the tag on, but the number and letters on the tag itself, you could probably see with binoculars, or if you had some sort of telescope, that type of thing, that you could be looking in an area that you've gone back to, and you could maybe see the tag, and you'd be able to know what the identifying information is. However, if you want something more sophisticated, collars or tags that track the organism, another way you can do that is radio tracking. Wildlife radio tracking is another method. And what happens is you put the tracking system onto the animal, and then you can use frequencies like a radio transmitter frequency, and you can study the movements of the animals, behaviors, population sizes. And you wouldn't need to, be visibly able to see them. You would just have to have a receiver to be able to track the signal. So that was a second way, the wildlife radio tracking. Okay, we come to our first set of questions for the second part of lesson 19. Remember, there's two sets of questions in lesson 19. So we're on page 167. We're doing the check your understanding questions at the top of page 167. And question 1 says, what would be the best method of sampling animals that are awake mainly at night? Remember, we just talked about that. That's called nocturnal animals. So what's the best method of sampling animals that are nocturnal? Okay, you can't, not going to be out there in the field when you go walking out in the middle of the day. So what can you do? Number two, what is number one was worth, on page 167, is worth 1 mark. And question two, what is wildlife radio tagging? What does it allow researchers to study? So with that one, you have to give me a description of what wildlife radio tagging is, and then what does it allow researchers to study? So that's 2 marks for that question. Okay, the next type of measuring and monitoring technology, of course, is from space. From space. This is called Earth observation technology, and we use it to measure and monitor biodiversity. So it includes satellites, global positioning systems, right, GPS, and they collect computerized images and data from space. And so what they're able to do is use GPS to track animals. So what they do is they put on a collar, somehow attach a radio receiver onto the animal you're trying to track and to study. And then that receiver picks up signals from satellites, and they calculate and store the animal's locations. And then you get your data. It can get all kinds of data actually about the planet in general, about oceans, about wildlife, about forests, everything about that. So it uses satellite images like the one in figure 6.12 to measure and monitor Canada's forests, for instance. And they're showing you an example there, figure 6.11, of a receiver that's trying to measure where the animals are. So we're just going to go over here for a minute. Sorry. And we're just going to go back to our website, and we're going to look at methods of tracking animals. Okay. Let's say scientific methods. Okay, and there's lots. Okay, so here's lots here. Right, there's some really good stuff. Okay, so there's a really good example there. Oops. I've got the wrong one. Just a sec. I shared. There we go. I got the wrong page shared there. Alrighty. There we go. So in this picture here, you can see a crocodile, and it's a saltwater crocodile, and it has a GPS satellite transmitter on top of it. You can see it right there on its back. And that transmitter they use to be able to track where the crocodile is and what it's doing. And you can use that using GPS. These are sea turtle eggs being laid by the mother. And we can use those in stable isotope analysis, another testing method. There is a wolf in Yellowstone National Park. You can see it has the collar. It has the radio collar that you can use to be able to track the wolf. There's somebody with a mountain lion has a radio collar somewhere, and they're using it to track the mountain lion that has the radio collar. This one here is got a monarch butterfly, and it's been tagged, and a plastic sticker is placed on the butterfly, and it's used to study migration patterns of monarchs, which has been interrupted by global warming, climate change, etc. You'll see there that that little brush-tailed rock wallaby, that would be in Australia, or that part of the world anyway. And you can see it has a little radio tracking collar on. And there's our again, our crocodile. So that's satellite tracking. All right, we're also going to look at EO track technology. I believe there's probably lots of different. Okay, so it says I don't know if these are specifically, let's say for tracking animals. Maybe that'll help us be more specific with it. There we go. Look at that. There's a lion and it has a collar on, and it's showing the that it's being tracked by EO. Okay, that's Earth observation technology. And it says here that GPS receivers on the animal pick up signals from orbiting satellites to calculate its location. The data can be stored or transmitted remotely via satellite or cellular networks. Let's just go to this site for a minute and let's see some of these examples of EO tracking wildlife. Okay, so it says here, as mankind grows, sorry. As mankind grows and expands their civilization, nature and animals are becoming endangered. In accordance with nature's guidelines, each and every living creature on this planet is vital and has their own significant ecosystem. Also, people use certain animals to make medicine and treatments to cure illnesses. Okay, so in this particular picture, you can see that they're tracking, to say, I believe it's birds. So here they go here. What data is acquired using GPS? Position of the tracked animal to determine the environment and the living conditions. Mortality sensing unit. If the animal hasn't moved, let's say in 4 hours, it could, let you know. Temperature sensing, detecting the body heat. LCD display, a microcontroller, and you can look for the exact physical location of the animal. Okay, so that's it. There was some really great pictures there, but not sure what happened to them. Let's go back for a minute and see if we can find. Let's go to this one here. Nope, it's the same website. Okay, maybe if I just go over here and yeah. So there we're going to have the lion. Here we go, all sorts. You have a little dog, but I believe it's a dog. We've got a tiger. There's all sorts here, birds, different kinds, cheetahs, right? These are all different types of animals that have these different ways of tracking so that we keep track of what the animal is, where they are, and other information like that. Okie dokie. So let's go back to our textbook. And okie dokie. Textbook. And here, oops, just a sec. There we go. Hopefully you can see that on the screen. Right now. And we're now going over to the review questions. Alrighty, we're over on page 169. And we're on lesson 19 questions. So I just want to go back and remind us of how this works. If we can go down and just give me a second to find the questions are on page 12 of 15. And we're going to do, all right, so if you remember last night, there they are. Lesson 19 key questions are on page 12 of 15. So last night, if you recall, we began the questions in the textbook on page 157. Those were from section 6.1 of the textbook. And we did check your understanding questions at page 157 and page 159. All right. And then we did review questions on page 161. And those were from last night, section 6.1. Tonight we're on section 6.2. So we're going to switch and we're going to go over to review questions on page 169. So, okay, and there they are listed right there at the bottom of page 12 of 15. So there's two sets of questions for lesson 19. Okay, but it's all within lesson 19. So lesson 19 is going to cover two sections of the textbook. Alrighty. Give me 1 second and I'm going to go over and we're going to go to those textbook questions right now. Alrighty, there they are, page 6.2 review questions. The first question is question number 1. What is the difference between how living things are sampled in a quadrant sampling and a line transect sampling? So you can just describe those two different, the two different ones, and that will be fine with 1 mark. Page 169, question number 2. Describe a situation where you choose a line transect sampling over quadrant sampling. All right, I'm not 100% sure that one was as clear. If you you go on into there and have a little read again, each sampling has like a different why you would use it. When you're looking at a quadrant sampling, you're looking at the whole area, right? Line sampling, you're looking at just the population living along that line. So when you're looking at more of the overall area, you're going to be looking more at the quadrant sampling technique. Whereas with a line sampling, you're actually looking at a specific area, right? If you look at how you want to compare the species living at one part of the forest to the other, a line transect sampling would be a better one, right? Because you could go along the line and it would be comparative, which is what you want. All right, question 4. Describe the difference between passive and active sampling. And we've discussed that, so that's 1 mark. We've discussed the difference with passive, right? You're trying not to disturb, you're being passive. Passive means to not be acting, right? To not disturb. And active, of course, we know what active means. Alrighty. Question number 8. How are aquatic dip nets and mist nets similar and different? Okay, so that question is worth 2 marks. So go back in. We talked about aquatic and dip nets. There's actually a nice picture at the bottom of page 165. It's showing the aquatic net and it's showing the dip net. You could talk about what it is you're trying to catch with those nets. You could tell a little bit about how the nets are made. Okay. And that might help you out with some different, how they're the same and how they're different. So 2 marks for question 8. Question 9, in what type of situations would helicopters be used to capture animals? So you can talk about what, helicopters, what they would use to capture, right? What kind of animals would be good? I don't know that we really looked. There might be some really good. Let's go to our website here and check and see if there's some good helicopter tracking. Oh, helicopter capture of animals. Oh, that's what it is. Okay. Okay, let's have a quick look at this. Actually, this is good. Here's a nice article from 2024. It's giving it a second to load up. So they're showing, it's maybe not this one. Here we go. Okay, so a net gun, right? Okay, so we're on the site. It says helicopter capture of animals is a method used by wildlife professionals for research, conservation, and management by allowing them to swiftly access and capture animals in remote or large areas. So they use darting, net gunning, herding animals to a specific location. Then the ground teams move in. They are often to place tracking devices. So there you go. And put on GPS collars, collect health samples, or move animals for relocation. Okay, so the purposes of helicopter capture then, researching and monitoring, attaching GPS collars, collecting data for studying animal movement, survival rates, and habitat use, conservation, catching and relocating endangered species, moving overpopulated herds to new areas, health and veterinary care, swiftly accessing animals for health assessments, collecting blood for disease at testing and administering treatments, and gathering information on populations, which is what we've been talking the most about, such as pregnancy rates in females, managing herds that may cause damage, and of course, relocation. The methods to do this are darting. A helicopter pilot maneuvers to a specific animal, allowing a vet or trained professional to shoot a tranquilizer dart. You can use netting. A helicopter uses a large net gun to capture specific animals like white-tailed deer. Herding, use helicopters to herd the animals, and then you have people on the ground that can capture them. And ground team support, a helicopter can track a darted animal until goes to sleep, and then you tell the crew where to get to. Okay, so those are all methods and techniques. And so one of the things it asked in question 9 is what types of situations would helicopters be used to capture animals? And here it's saying minimize stress, safety of the team and animals, efficiency, and expertise. Good stuff. Okay, let's go back to our questions. We are on question number 11. How can satellites be used to help track larger animals? So that is worth 2 marks. So you want to talk about how you have to put on the collars, right? Or a tag of some sort, and how you have to then keep track of them, right, with the satellite. And when we think about GPS, that's satellites, right? Think about GPS. How do we use GPS to track anything nowadays? I can track my kids with their cell phones because I have the Find app on my cell phone. And so I can go in and I can see where my children are and be able to track where they are and um, be able to locate them. And I suppose that's pretty much what we're doing when we're using satellites. Okay, question 13. Suppose you were asked to determine the population of polar bears in northern Canada. What technology would you use? Explain your answer. So you're going to get 1 mark for that question for saying what technology you'd use, and you're going to get 1 mark for that question to explain why you would use that technology. So if you pick a technology, I'm going to guess you're not going to say quadrant sampling. I'm going to guess that. Um, so, but, or line sampling for that matter. But whatever one you pick, all you have to do is support your answer by letting me know why you picked that form of sampling, and that was worth 2 marks. All right, we are done lesson 19. Next class, we are going to begin lesson 20. Lesson 20 is like lesson 19 in that it has different parts to it. So we're going to take, and it's 3 lessons in the textbook, but I've cut back on the questions, and so it's down. It's 1 lesson in the booklet, lesson 20. And that's where we'll start in our next class. Thank you so much for joining me tonight, and we will be back on for the next class to talk about lesson 20. Thank you. Good night.